Hey everybody, quick audio check before I get going so everything is all nice. So far it seems like it's okay. Let's bring in the music, courtesy of JX3D again. Testing, testing, testing. I think we're okay. Just verifying real quick. Getting an echo because I'm using my phone to test audio. Sorry about that. Okay, I think we're okay now. I'm going to have to turn one of these down a tad. I think I was a little too hot on my own mic here. Anyway, anyone's on the stream, you can let me know if it's off, but it seems to be okay for now, for me anyway. I'm going to periodically check one more time here, or maybe one other time later just to make sure nothing changes. Alright, seems okay to me now, so I'll leave it there instead of fiddling with it too long. Nobody wants to hear a bunch of tests. Alright, so what we got going on here is some samples from Boronda Lake and Foothills Park. I was talking to one of the current teachers there that I've known for a long time, still works there, and we were talking about some stuff I've done on my channel and other stuff I did at the Baylands when I worked out there, and she had mentioned they never got any good indication of the critters that are in Boronda Lake other than the real obvious stuff like the birds, if you saw my video from earlier tonight on my synthetic channel. And so I thought, oh, let's get some samples while I'm driving through the area. And that's when I shot that video. I had to upload it as a premiere because live streaming wasn't working. It was too, just basically no signal at all. Couldn't make a phone call, couldn't do anything. So I recorded a local video and just did a premiere about half an hour ago, 20 minutes ago, something like that on my other channel. So, hopefully that brought you here. If not, you're already here for the streams I already do here. Actually, I did a check. It's been at least 2 months, 3 months, probably about 3 months since I've done a stream here. So just been so busy and lots of stuff going on and traveling and all that stuff. Just haven't had the time. But now I have a good excuse because I want to cover this content for the teacher. And if I get enough decent footage here, find some nice critters to catalog, I can make an identification sheet out of it and even put some videos together for her. I'll just strip the audio out so they don't have to have the music and my yakking about random stuff. Anyway, so this is a sample right by the dock that's in the video where where the cormorants were sunning themselves. And there's a bunch of bird fecal matter on the dock, which is always wonderful. But they wanted to keep it natural somewhat there, so they don't clean up once in a while, it looks like. So I reached over the edge of the dock and got a decent sample of the water just kind of right by the dock near surface level. Since a lot of the critters and maybe even single-cell plants will be hovering near the surface, they're not going to be deep in the water. I can go back with a longer pole and actually go further down. Maybe you can get a little bit of mud in there and a little bit of the algae. If you saw the video, there's a bushy algae kind of clumping a couple feet under the surface, so I can even use some snips and basically grab a piece of that and whatever's attached to it. This is just a quick and dirty, get a sample at the dock and then get a sample at the shore with some little kind of grasses and stuff that were sticking in the water that had some, looks like they had some growth on it. So hopefully I'll find something here. So far, I'm not finding anything on this guy. This is a piece of, looks like probably dead animal tissue with all kinds of fibers here. This couple of these weird lines on the top are from, I got some water on top of the cover slip by accident. I was kind of sloppy when I put this together. But, so we have mostly fibers here. Fibers of stuff. I'm using dark field right now, so it shows up much more contrasty. Again, lots of fibers, no movement. Nothing exciting whatsoever. Got some more some tissue there, some kind, more filaments, plant fibers, stuff like that. Oh, there's a little tiny flagellate down there, looks like, dancing around that fiber. Oh, no, wait a minute. There was something out of focus. Sorry, my bad. It looked like it was moving, but it was the parallax from my eyepieces. There's that clump again. Let's get a little more brightness here. A little more brightness to the field there. Looking for other particles. I'm not using my fancy digital camera tonight, just a USB camera. It's quick and dirty, gotta have time to set everything up just right, but it's always better when I use my nice digital camera with the adapter. If you saw my last few videos, maybe not the last one, but a few before it, you can definitely tell the quality difference of the images, the brightness, the colors, clarity. Okay, so nothing on this slide is a wash. Now it could also be I just got a boring part of the thing, so I didn't really mix it up too much. I just kind of grabbed it out of the vessel. So what I can do real quick is poor man's centrifuge. Hey Davey, what's up? Enjoy JX3D's music in the background. It always seems to go well with the stream, and I don't have that much ambient type content at this point. Just a few tracks here and there that would really fit, so it's just nice to give JX3D some extra promotion. go to his Bandcamp and you can buy this stuff. Anyway, so I'm doing poor man's spinning of the vial to kind of drag the critters more to one side than the other of the thing, force them toward the bottom. And if I didn't make this too deep, I can reach down there and get a better sample of these guys. Potentially, not guaranteed. Let me wipe this off. These slides and cover slips always come out dusty. So you can't just pull them out of the box and use them. You have to give them a quick wipe. The manufacturing process just leaves some crap on them. Davey, let me know if the audio bounce is still okay with the voice and the music. I did a quick test or two before I started, but I wasn't exhaustive by any means. So. Alright, let's get this. Cover slip is filthy. It's brand new and it's already kind of smudgy. Always have to wipe this shit down before you use it. Something they don't tell you when you buy this stuff. You will have to clean every piece before you use it. Alright, let's try this one. This one's been spun a little bit by hand. Obviously, I have to buy a proper centrifuge to make this a little bit better, but... Okay, so there's just some capillary action there, water moving around. More fibers. More fibers. Ooh. No, more fibers. Thought that might be an exoskeleton of something, but it wasn't. A whole lot of debris in the water, though. I'll tell you that. Focus through another layer. Oh, that's the top of the cover slip. Let's go. Yeah. Nothing yet. Just lots of fibers and such. Pretty boring sample. I know the stuff at the shore will be nice because I got some bits of plant material, grass and stuff with it that has most likely has some stuff clinging to it. And some stuff like it started to decay a little bit, so there's probably lots of stuff they're trying to eat it. So yeah, this is a wash. I might at the end do one more sample from it, but it could have just been a really boring part of the water where I grabbed it. Water column was not very exciting right there. So I'm going to, and you know, there's plenty of color to the water. There's definitely, you can see all the little fragments in there, filaments and little bits. This one, I didn't get the dissecting scope ready tonight. I was just doing the compound scope, but there's definitely some fragments and stuff. I may, if I feel up to it, maybe tomorrow. These samples are usually pretty good in the fridge for a day or two, so maybe tomorrow night I might do another one of these before I toss them, because you never know what you're going to find. So we'll mix that around a little bit. Let's get a sample here. Let's grab clean the slide again, because again they don't come in all perfectly clean. I think if you pay a lot more money for your slides, you get them from a special place. They may ensure that they're perfectly clean, ready to go in a lab situation where you don't times are wasting. my sample. So we got here, this was from the shore. And much of the same. Particles everywhere, but no action. Oh, wait a minute. There we go. That's a fast one there. Holy smokes. That might be... Oh, that's nice. What is that guy? He's zigzagging like those silly alien videos you see online. That is cool. Let me, before I change eyepieces, let me just do a digital zoom real quick and see. I can cheat it real quick. Alright, where'd you go? There it is. Zigzag, zigzag. That's one of the fastest things I've ever seen on a slide. There's like a air bubble trapped in some debris there. Looks cool, but it's nothing special. Some kind of colorful junk there. Nothing remarkable except for that little fast guy. Oh, look at that. Got a piece of the grass in here. Interesting. Let me bring up the brightness on that. Nothing on it that I can tell. And you got this air bubble here. I gotta find that little fast dude. That thing was cool. I'm seeing tons of tiny little flagellates. If you look very carefully, you see little particles dancing. Oh, there's something moving there. Oh, that's that same guy that was... There he is. Now he's whipping around. Alright, I gotta go higher power. This is too cool to miss. Let me get this adjusted. Got some oblique lighting now. Let me fix that a little bit. Let's go back to bright field for a second here. Here's your standard microscope view. So there's another small one kind of bouncing around, but it's not the zigzaggy fast one. I'm having trouble finding that guy again. Where'd it go? Adjust the contrast here a little bit. Okay. All kinds of fragments of stuff. clumps of cells. It's a nice clump of cells there. Let me get my this view is not centered properly. That's pretty good right there. A little clump of cells. Give a little contrast adjustment again. There's a little flagellate poking around it. See little particles with that zigzaggy movement tumbling around? Those are flagellates. They have long flagellum that makes them tumble like that. They're not turn on a dime direct swimmers like ciliates are. But they're just kind of going around that clump of cells and not doing anything with it. Alright, I gotta find that fast one. Where did that go? There's another one. See a little ball, little dark guy tumbling there. A lot easier to see when it's. Let me go back to the dark field again. This is a little bit more of oblique lighting. It's not quite dark field. It's a little more 3D looking. Get a little. There we go. Fix that condenser view a little bit. Okay. So now you can really see now the little white things cruising around instead of little black things. But they're going up and down in the liquid, so I have to constantly focus up and down to catch them. Very quick. But they're not as quick as the other guy was jetting around. I have no idea where that thing went. It's really cool looking. I have to scour. Find it again. It's kind of an ovoid shape. That's some of the cool effect you get from oblique lighting. It makes it look very 3D. There's the air bubble in the middle there. Or water bubble with the... Let me fix... Got some weird artifact on my field of view here with the... Alright, let me make sure this is correct. Fix that a little bit there. That's more like what I'm seeing in the eyepiece there. Yep. Okay. Cool. Now the trick is to find that one character. Where the hell did it go? I might have to go to low power again to find it. Fix the dark field a little bit more. Okay, where did that dude go? He was fast. No movement. No movement. Seeing it down here. It was hanging out toward the bottom earlier. Nothing, nothing, nothing. Now, unfortunately, it can get sometimes beyond the cover slip a little bit. Well, this is actually a barrier. It has to stay inside this water jacket here. So it's definitely not down here, below this line. It looked a lot like this blob right here, but this is just something else. Let me fix this focus here. Ah. All right, there we go. There's Noah's little flagellates cruising by on the right side of the screen. But I can't find the fast one. There's the guy in the middle again. Ooh, there's another spherical thing that looks kind of green. Let's go a little bit higher. This is 400 power. What's that guy? Some little single cell planty thing. Very green. Get more light to that so you can see the color better. Let me do a cheat because I don't want to go any closer. I have to go to an oil lens, and let me basically blow this up a little bit more. You can see a little bit better there. Got a greenish tint to it. You can see a little. It's almost like a. Oh damn! What was that thing I found not long ago? They have this what seems like a gelatinous barrier around them, which gives it that little ring around the outside. And it's like a protective layer. It's not the cell membrane of the cell wall. I forgot. It was a specific organism I found in my bird bath. It looked like this single cell plant. I have to go back through my streams and my notes and stuff to find it. It's been months since I looked at it. But interesting. Let's go back out because everything is too far away. Too far out of the field to actually find anything else. So let's go back to just that. Keep looking for the fast. There's another little flagellate cruising around. Too hard to identify something that small that moves that fast. But nonetheless, there is some activity. Just can't find that really cool fast one. Oh, there it is. It slowed down a little bit. Yes, let me zoom in now. Fix this. There you are. You're the fast guy. put a little too much liquid on the slide, so it's bouncing around in the matrix there a little bit. So I don't touch anything. Maybe a little less wiggly. So you can see it's spinning around. Very, very active, but it may just go ping and take off on the screen pretty soon. Unfortunately, this is my 400x equivalent objective, and I don't like. I made a mistake and put an oil, the original, and this one hasn't quite matched since I replaced it. So I need to get a proper replacement because this one has a little bit of a haze to it. It's not a perfect eyepiece or perfect objective. Well, at least you can see the guy up close who was cruising around all crazy. He stopped going so fast now. He's just kind of slowly moving around. I'm pretty sure that's what that was, because you can see some cilia. It's not quite as wiggly as the other ones. No, wait a minute. No, it is pretty wiggly, actually. I think it's just a larger flagellate. So this isn't the one that was cruising around all crazy. Because the other one had some, like a tuft of cilia that it was using to propel itself all crazy around the screen. This is just another flagellate. Interesting though. So a little more contrast here. Yeah, so that all that wiggling is, I thought I was bumping the scope too much. Sometimes I put too much water in the slide, and any slight touch of the scope makes it all jittery, but this guy's doing his own thing. I didn't touch the scope for a while. He's definitely a wiggly, kind of sloppy version of swimming around. Cool though. It's a nice little specimen. Not sure if I can identify it from all this. It's not the best image, but you can see some contents inside. But it's very small. This is 400 power, so human cells at this magnification would be quite large. So this is a little guy. Plus I'm also digital zooming it on top of the eyepiece equivalent, so it's actually more than 400 power on the screen. Yeah, this would have been a good candidate for my better camera. This camera's not. A combination of this objective and this camera, not quite getting the sharpest image I want to get. But yeah, it's cool. Let me go back out. I gotta find that other fast one. That thing was cool. I'll do it before the slide dries up. That's always the scary part. That might be the only time I find this guy in the water, and I'll lose my chance. Okay, so it's nowhere in this search perimeter that I can do. I'm seeing a lot of stuff is about the same size as it, but nothing is moving anymore like that. It could have already expired from the heat of the lamp, or just some of these guys don't live very long, so. Seeing Lots of tiny stuff in here, but nothing more like that other cool. Be nice if it was hanging around some stuff and maybe, or just park. Sometimes things park around some debris looking for stuff to ingest. And I'm not seeing it. I think I got the only shots of it I'm gonna get. But hey, that was pretty cool for the first slide. That was already way better than the others, the other samples I got. So let's try another one from the same vial. Hey, what's up, Aussie Head? How you doing? Doing some water samples for a friend at the museum and zoo that I used to work at. She's a teacher there. She wanted to see what was one of the local parks. They do some programs there in the city of Palo Alto, up in the hills. If you saw my earlier video, I did a premiere event and sent that a little bit ago. Because I couldn't live stream today from there. There's zero phone signal at the site. I had no idea. I forgot it was so remote enough that it didn't have a cell phone signal. It's only like 15 minutes from town, in Palo Alto up in the hills. They must not have enough cell tower coverage or something up there. So anyway, I shot that video and then I did a premiere about an hour ago at this point. And then I put a link in that video for this video. So once people saw where I got the footage from and saw the birds and some other scenery, and then they come over here and see what's in the water. And that's what we're doing now, is to see what I could find. So far I found some really cool little zippy ciliate kind of thing. But it kind of came and went. I couldn't find it after a couple of attempts. And I wanted to do a closer shot with digital zoom and also an objective, a higher objective. And it just didn't get a chance to get it before it was gone. So this is sample number 2 from that same vial. This is taken from the shore at that lake where there's a bunch of grass, debris, and other stuff for things to cling on to. Let's see what we can find here. Let's do my standard search pattern and see what we can see. It's a little dark. Let's fix that. Okay. Lots of debris and filaments and fragments of plant material as usual. That looks like it could be something, but it's not moving. Let me zoom in a little bit. Yeah, it's just a cell of something. Who knows what it's from? Nothing exciting. Just a little oblique lighting, that's always nice. I love the 3D effect. Got some, oh, I see some moving? No? No. Nothing moving. you guys don't already see the notices, JX3D is covering for Ranzi tomorrow, or tomorrow for me anyway. And also doing a senior synth stream the usual time. Basically noon my time, so 8 o'clock UK time, with a bunch of guests. We're going to talk about unusual instruments, stuff with strange interfaces and control. Some not that strange, and most of you have already heard of most of them. That's an air bubble there. That's a piece of something. What is that? Probably nothing, but I'll zoom in anyway, just curious what the hell it is. Excuse me. Oh, I lost it. Where'd it go? Yeah, some little piece of something. It's kind of interesting, but nondescript tissue. Pieces of various debris, all kinds of unusual shapes, but nothing significant. More tissue from something. Probably some random animal tissue. Could be some of the stuff could be, it's right out of the water with all the fishes and turtles and other stuff, and the stuff just floating around. It could be, a lot of this could be just, digested animals, fecal matter, stuff like that. Nothing stands out as stuff I'm normally looking for. So other than that one guy in the last slide, this is pretty much boring as well, unfortunately. I think I covered the whole slide already. So some little filaments there. There's like some like blue-green algae filaments. Those little hair looks like little hairs. If I change the contrast, you'll see what I'm talking about. Like on the left of that long rod there, those little white-looking hair things. Those are basically really, really small blue-green algae filaments. Nothing too remarkable. I don't get any diatoms even. It's interesting. Yeah. Pretty dull. Alright. I don't waste too much time. On to the next one. Let's make another one. I'll try and stir up the water a little more this time. Maybe I'll get some stuff settling out. Could be that the lake is just kind of, other than the birds in there, relatively uninteresting. Although all the birds coming and going should be bringing stuff on their feet and in their bills and stuff. There should be all kinds of stuff in there, but I have to go a very specific part of the water, or at a certain depth. So I'll have to go back and get a couple different depths. I got only stuff at the surface or at the edge. I should try some more what you call benthic stuff on the bottom in the actual mud, or further down from the surface, just suspended, like some things that don't like so much strong light might be in there. So alright, just stirred up the water a bit. There's a lot of suspension, dark particles in the water. See if that helps. Like I said, I have samples are good for at least another day or two, so I'll probably also do another one of these. Give it another try in a day or so, and then we'll also use the dissecting scope with some of the grass fragments and look at maybe there's some other things on the plant fragments themselves. But this is part of the fun of this whole exploration thing. You never know what you're going to find. I'm not culturing anything. Some people do streams based on cultures they either bought or they have. And so you always know what you can find. You can count on it. But these are all just random stuff from the wild. No, so there's a nice blue-green algae there. Probably a Solatoria, maybe. See if I can get a closer shot of that. It looks like Anabina. Anabina or something like that. See, it's just a little line. If I get even closer, you'll see the segmented nature of it. There's individual cells in a colony. There you go. Do more light. So it could be any number of blue-green algae. We can see it's actually moving. They are motile. They have flagella and they can move. They have to because as a photosynthetic organism, as a colony, it has to be able to go find where the light is or get away from the light when it's too strong. So it looks kind of creepy, like a very slow worm, but it's actually a colonial algae cooperating together to move about. So this one's actually moving quite a bit. That's kind of cool. So yes, there's plants like this can move on their own. They're not stuck on the ground or planted like a holdfast of kelp in the ocean. They're actually free-swimming little colonies or single-celled plants. And in this case, at this size and this structure, it's not even a plant, it's a blue-green algae, so it's related to bacteria. It's very primitive. Representative of some of the earliest organisms on the planet. Plants and animals learn to be colonial, and then over millions of years of specialization and natural selection and some mutations that were actually good mutations, they became even more complex organisms. And eventually you got to us after a long time. As you guys already know, most likely, if you put human civilization, the entire human civilization on a calendar of geological time, we're like the last 100 seconds of the last month of the year. Our entire existence. 200,000 years of Homo sapiens. So, it's pretty crazy. Puts it in perspective. People say, oh, this stuff can't possibly happen. These time scales you're talking about. I mean, evolution is a very slow process. But you can have it happen over a shorter period of time with environmental pressures, various predation pressures. There's a thing called adaptive radiation where, like the honey creepers in Hawaii. Hawaii is a volcanic island chain formed long after life was formed on other continents. So how the hell did the animals get there if it was brand new land from nothing? They either flew there, blown there in a storm, rafted there, until later when humans actually got there and then brought other animals there. That's the only way they could have gotten there. And once they got there, if they survived as a breeding pair or a small colony or something like that, then they would continue to specialize isolation on the island and adaptive pressures and other kinds of stuff. And adaptive radiation allows the Hawaiian honeycreeper to, and they can check this genetics as well. This is not just visual stuff. The relatedness of all the Hawaiian honeycreepers. They look quite different, but they're all from like a parent group of Hawaiian honeycreepers. And over time, they had adaptive radiation throughout the areas that they populated and had isolation. And again, other pressures, the food source changed, the temperatures in some, and humidity in some areas changed the way they look, changed their sizes, changed the bill size, bill shape. They started specializing in different things. Same thing with the Darwin finches on Galapagos Island. They're the Geospiza genus. They have, I forgot now, it's something like 10 different species there, 8 different species, whatever it is. And some have massive bills and some have tiny little bills, and they all fill the niche, just like we have birds here filling a niche. You have the little, like little bush tits and wren tits and little kind of beep, 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 beep birds all hopping through the leaves with tiny little bills picking insects and other things off of leaves. And then you have other birds like a woodpecker that can drill holes and pick away at stuff with a chiseling bill. And then you have other birds that can crush things with a big bill, like crushing like large seeds and stuff. So it's just all this different variation. It's really interesting stuff. But look at that thing move. It's like a giant, slow submarine. And again, I don't remember what it is. It might be Anabina. I don't think it's Ulothrix. Ulothrix is a lot bigger. But look how it's also flexible, too. As it's moving, it kind of undulates a little bit. So it acts like a single organism, even though it's a multicellular colony. So they have this communication between the cells to organize. Just like you get in a slime mold. When a slime mold comes together from individual cells and suddenly starts forming connections and communicating, just like your cells in your body communicate as one. They cooperate. This music's perfect for this because it's kind of a marching, kind of a slowly marching music as it marches on by. A little more light to this thing. That's probably the best it's going to get. Take it slightly out of focus on the edge so you can get the individual cell barriers there, the individual partitions. No, it's not a worm. It's worm-like in its motion, but it's individual what's called cyanobacteria, is blue-green algae that are related to bacteria that can photosynthesize. And instead of living individually, they attach together as a colony and communicate and cooperate. And they can beat their flagella in unison and move. So that the structures are very tiny. It's very hard to zoom in on them to see any kind of motion like you do on like a paramecium with the cilia beating, or like a euglena with a large flagellum that you can see very clearly whipping around. These guys are very, very tiny structures because I'm already at, this is 400 power. This is a very small, thin thread, but you couldn't miss it because it had a nice color to it. It's a greenish tint. So, and now it's just starting to move. It's probably getting a little too much light from this incandescent bulb in the microscope, so it's actually moving away from it. It's probably not too happy with it right now, but I have no choice if I want to get a good image. So it's a little bit of heat and a little bit of extreme light, but this is the only way you can make this thing work. I didn't want to get the LED lights that I have in the dissecting scope because it's a colder light and the colors are less natural. But the cooler light does make the specimens a little happier. So you sacrifice drying out the specimen sooner and maybe making them a little bit unhappy versus having realistic colors to your specimens. It's definitely a trade-off. But even sitting idle, a slide will evaporate just from the temperature in the room. So I've shot a bunch of these things before. This isn't anything new or that unique or whatever, but it's fun to see them when you can. And when they move this much, it's like really dramatic, you know. It's people don't think of plants or plant-like things like the cyanobacterian stuff or these colonial threads actually moving like this. But diatoms do the same thing. The single-cell plants inside of a glass shell, they also move around, but they don't have this flexibility. They're rigid. So it's fun that this thing is very flexible as it's moving. You can see the tip is bending, and the whole body has an undulation to it. Let's go back to the front, or what is currently the front as it's moving that way. So if you were to speed this up and make it a little more extreme in movement, it's almost like a very tiny worm-like thing. There are some small worms that move slowly like this, and there's some really fast ones like nematodes that just whip around all crazy. Now it's going backward. So something is giving the signal, just like you have your train and the caboose. Something is deciding to go one way, and then suddenly it's like having the engine and the caboose changing roles constantly. I'm going first. No, I'm going first. So totally out of focus, you get the whole shape and some nice focus on the front part, and you get a little bit of a halo on the edge, and then focus through a little bit more and you get more of the cell wall structure in between the segments there. It's kind of a happy medium right there. Now it's going the other way. Pretty cool. Let this go a little bit longer because it's just fun to watch. And I'll poke around the slide again before it starts to dry up. And I'll make maybe a couple more mounts. It's getting late for me, and I do have to take a nap before the Ramsey replacement show starts up. I don't want to be totally out of it. I've been going for about an hour. That's decent. I can go for a little while longer. I gotta get some rest. A long day. But yeah, it's still going, still moving. I should put JX3D's Bandcamp link in here. People watching back. Oh, now it's coming back. It left and now it's coming back. It's like and it's perfectly placed too. It hasn't changed overall placement. It's just going diagonally across each time. It hasn't really shifted. I haven't had to move anything. It's funny. I'll give a little more brightness to this maybe. Yeah, that's about as bright as it's gonna go unless I tweak the diaphragm, and I don't want to do that. It's like a screensaver. Sit here and just chill out. I'll let you do its business. Nice thing is it's not moving in the Z plane very much. I can stay focused on it, and it just kind of scoots in and out at the same depth. Whereas a lot of the swimmers, especially the little flagellates, are all crazy. Constantly go up and down and up and down. So you have to do X, Y, Z all at the same time. Makes it a very tough video game to follow them around. Alright, that's good for that. Let's go back out. It's fun. And for idea of scale, let's just pull back here. See that little thread there at the top? Let me bring it down again here. That's what we just looked at. That little thread right there. Not very big at all. Yeah, let me fix this there. Fix this a little bit better. There we go. So there you go. That's what we're looking at, that little guy. Not big at all. That's at 40 power. And we were looking at it at 400 power. So let's cruise around a little bit more. See what else we can find. And I'll make another slide. Slide's starting to crystallize and dry up now. It's not salt water, but there's still a lot of solutes in it. It'll start to grow crystals as the water dissipates. Yeah, it's odd. No diatoms anywhere. water intrusion there. See, it's moving. Unfortunately, I got some water on top of the cover slip again. Yeah, nothing. It's a nice filament. There's the edge of the cover slip. And not even anything outside of it. It's the same old junk. Alright, let's try one more. If it's halfway decent, I may do one more after that. Otherwise, I'm getting tired. Come on, slide. Now these slides are sticking together. Shit. It's almost like some moisture got in the case. These are all stuck together, man. What the hell? Alright, let's try. Start it up a little bit more. That might actually help. May not, or may not help. Depends on the sample. Let's get a cover slip on here. Or some people call it cover glass because it is made out of glass. We had cover slips in high school that were plastic because they didn't want you to cut yourself. It's very easy to break these and get a nasty cut because they're so thin. Alright. Search pattern. Get all that movement as capillary action as everything settles out. Again, tons of tiny little, these little particles moving around the background there. If I give you a little more brightness, you'll see what I'm talking about. Little tiny dots that are moving on their own against the flow of the capillary action. Ooh, that is a nice one there, but it's really tiny also. Oh! I've got a spirilla in there, looks like. Oh, that is too cool. Hopefully it will show up in here. So it's a large bacteria that has a spiral shape. So I can find it here. Let's go. Why is that not behaving? You really need that kind of dark field look to get that effect properly. But I'll try a little bit of bright field first. See if I can find that guy again. That was really cool. They move like little corkscrews, and it's like a perfect shape to move through kind of gloppy environments and tissue, just like you would have a corkscrew go right through a cork. They can just twist right on through instead of getting stuck. If they're like a rod, it would be like having to put extra force to kind of dig through. But being a corkscrew shape, it's a really clever shape to move through things. I've actually watched them just kind of push their way through kind of slimy, gloppy debris before, just no effort. Whereas something like a paramecium would have to kind of go, just kind of nudge itself through there see it. See lots of debris, but nothing special. But it had that telltale thing where it kind of stops, and it goes back and forward, while it's spinning on its axis, just like a little corkscrew. And it was very large. For me to see it at that low power, it's a big one. Usually they're not that big. There's a whole bunch of debris there. Plant fragments. Yeah, I'm not seeing it. I have to go back to low power to find it again. Again, lots of junk, but nothing, nothing doing much, nothing. So far, several little tiny things that are very hard to see. Just the luck of the location, just sample's not so great. But you never know. If I kind of scraped the leaves a little bit, probably would have gotten some more stuff. So there's some more of those, what they call cyanobacteria, blue-green algae threads right there. There's one right there. Pretty long and has a kink in it. See what we can do here with the dark field. Gotta find that guy again. There's a fast one right there. Look at that thing going. Holy smokes. It's very wobbly, but it's got that classic flagellate swimming pattern. And looks greenish. It's probably a single cell plant with a flagellum. Boy, oh boy. It's flying. Oh, suddenly my frame rate dropped. What happened there? Looked a little choppy. Shouldn't be choppy like that. Let me see what my setting is in the yeah, it's the right format. Why it was choppy. It's pretty smooth when I scroll. Weird. That guy was pretty choppy when he swam by. Not supposed to be choppy like that. It's not a stream thing, it's actually it showed that way on the live feed coming to OBS before it even goes to YouTube. Again, I'm not seeing anything really remarkable. More of the same junk. Alright, we'll make one more slide. Can't go out like that. Gotta have a nice one to finish with. The reason I'm so persistent is on a sample I got out of the bay, San Francisco Bay, from the city of Tiburon, which is kind of a touristy place, lots of restaurants and stuff. It's a nice view. It's kind of north part of the bay, still inside of Golden Gate Bridge, and closer to Angel Island. Just got a random water sample in the bay there, and on one stream I found one single dinoflagellate, not a group, not a cluster, one, randomly out of several slides I made out of that one vial of water. And those are little critters that cause red tide. And it was stunning. If you go back and watch it, I actually made it the poster frame for the video. It looks like a little Death Star with a red eye spot. And it has this little waist. It has like a shell around it. And a waist that looks like the trench that you go down in the Death Star. And that's where the flagella kind of wrap around and come out of the shell. And it was just, I was studying it for a while. It was really, really cool. I've seen pictures of them before and photomicrographs of them from, scanning electron microscopes and all kinds of stuff. But to actually find one cell out of all these samples and then not see another one out of the whole batch and then not see anything I've collected anywhere around there since, that was pretty special. That's why I'm usually pretty tenacious about this stuff. I don't give up easily because you never know what you're going to find. Just the one time, just before you pack it in, you might find something really cool. So, just like everything else in life, stay positive, keep trying, and you never know what you're going to find. Patience definitely pays off. But now that I'm tired, I'm not going to be that patient. I got to maybe do one more and I got to get out of here. So, let's try one more. I stirred it up a little bit. Okay, let's see what we got here. Might be just more of the same. Might get a special little character in here. Alright. So more debris at the top. This is always a fun shot. You can see the surface tension of the water as it touches the edge of the cover slip. You can see a clear ramp as it tapers down to the glass below. Really cool. Probably have to change the light to see that better. You can see that when I change the light levels a little bit. The camera's not as forgiving. The eyepiece does a much better job than the camera does, but you get the basic idea. That looks like it could be, at a quick glance, it looks like a diatom, but I can see the segment, so it's a piece of the kind of blue-green algae, or some other green algae that's much larger than the other ones. Colonial algae. Ooh, what is that down there? It might be a tiny diatom. Very tiny. It's got the shape, it's got the color. very tiny. But maybe not on further inspection. Low power, sometimes stuff looks a little more interesting. Okay, this is off center again. There we go. Scan the bottom. There you go. A couple little fast guys going by. More flagellates. But again, impossible to chase. I mean, I can try, but it's... You end up getting, like, people probably get seasick watching this. I chase it like that, you know. Chase it around, chase it around. You're not going to get more detail out of it. You're just going to get to watch it swim. So, stuff like that, you have to either... I mean, I put these in the fridge to make them last longer. The animals will slow down. But another trick is you make the medium thicker. Now, as it is, at the scale they're at, the water is already quite thick. It's like swimming through supposedly equivalent to us trying to swim through glycerin or something a little bit thicker. But you can actually make it even thicker by making it more like glycerin. You add methylcellulose to the mix. And it's the same stuff they use for the slime in the movie Alien. Anytime you have a slimy, gloppy horror movie or sci-fi movie with like biological slime or, you know, squishy, disgusting bits of just, you know, it's usually just clear. It's not necessarily colored or bubbles and anything like that stuff. But that you can add. I have some mixed up in the fridge that I use once in a while. When stuff gets really crazy fast. The problem is once you add it to the slide after the fact, it takes a while to dissipate through diffusion. And on top of that, you just lost your specimen. As it gets washed away, you have to go find it again. Or you can take a sample you know has a lot of fast things on it, just from the collection vial that you have, and just add the methylcellulose ahead of time before you start scanning for stuff. And then you'll see stuff moving much slower, because a whole lot of work to swim through that. So there you go. Here's one spinning in circles. I love when these guys do that. Let's go a little closer. Spinning in circles. We like that. Then we can study them better. So sometimes ciliates will do this. So I mean, flagellates will do this. They'll just go in a circle like this, like they're going bonkers. Let's change it to bright field real quick, just so you can get the average view you would have in a standard boring scope setting. So you see the animal has a little bit of a color to it. I can do a digital zoom a little bit to cheat and not have to change the eyepiece. Or the, not I keep saying eyepiece, objective lens. And then you get a better view of this little guy. It's got a really interesting tumbling motion there. You can see how it kind of rolls over. I have to constantly focus up and down a little bit because it is changing Z depth as well as spinning around. You can see it has some structure to it. Almost like it has an eye spot. There's some like kind of a colored spot there. It's kind of orangey reddish. Oh yeah, very interesting. That weird tumbling thing it does is fun. It doesn't just spin, it does that weird kind of tumbling. And this might be similar to the one I saw earlier that was so fast. I just couldn't find one that slowed down. That one was going zoom, 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 jumping all over the screen on one of the earlier slides. Too crazy. That's at the bottom of the slide, so I can't make it. You can probably... There we go. Tumbling action is really odd. I usually see them just spin or swim away, but never this kind of tumbling thing. It's like it's a roll. It spins and it does a roll and then it spins again. So it has a large white vesicle. It's not just a typical organelle, it's probably a contractile vacuole, that white thing on the edge of the cell. You can see it looks almost like a notch cut out of it because it's so close to the edge of the cell. I think that's a contractile vacuole, how they control their water pressure. What is it called? Osmoregularity. Not quite as important in fresh water as it is salt water, but because the internal cell is salty, as you know, compared to the outside water, you still need to have some kind of a thing to avoid getting filled with water and exploding. So what happens is you see when these things die, the contractile vacuoles stop working and they start puffing up and they eventually pop. Because some animals have pretty close salt concentration to the ocean and some less so. So in the ocean, if it's a freshwater critter and you put it in salt water, it's not going to be too happy, but it can probably deal for a while. Unlike if you did it with a fish. A fish would not be happy at all, but these things are sometimes a little bit more adaptable. Depends on the specimen, but there is a bigger concentration difference obviously in the freshwater to the inside of the cell. But some of these critters evolved in freshwater, so they're used to dealing with it. But once that vacuole stops working, then as it dies, then it will definitely pop. Yeah, you can really see that big white structure at the edge of the cell. I have footage of the paramecium actually pumping its contractile vacuoles, and it's really extreme. I mean, it has this like kind of a star shape as it's pumping, and you can't miss it. It's really dramatic. That's pretty cool. So this is not one I know, but maybe a few stills from this video will help me track it down. It's not the best image, and it is moving a lot, so each still is going to be a little bit blurry. But at least it's not flying all over the screen. I can actually enjoy it for a while and not have to chase it down. Well, I'm I'm losing it now. I'm getting pretty tired, so I'm going to call it here, I think. Let me zoom out a little bit so I can keep it in the frame a little bit more. Can enjoy it. Get the hand out of there. Watch some more antics before I call it. So, some success. Found a couple cool critters. So not bad. But yeah, some of the slides are totally dead. There's not much going on at all. So, seems like the first slide and the last slide were the best. The two different areas of the pond. Or lake, as they call it. It's a very small lake. It's more like a big pond, but they call it lake. So, anyway, I'm going to call it there. So, thanks everyone for hanging in there. I would stay longer, but I'm just beat. And I got to get some sleep before the Ramsey replacement show. So, thanks everybody for hanging out. And look for more stuff. I'll start doing these more often. Just took a couple month break, maybe 3-4 months. And I may do another one tomorrow night or maybe even Sunday night, depending when I have time to do another attempt, maybe find some more cool things or better shots of the same things. So I'm going to call it there. So thanks again, everyone, for dropping by. And one last shot of this guy before he goes away. And go out there and enjoy some nature. You don't have to have a microscope. You can use a hand lens, magnifying glass, turn binoculars around and put things very close to your hand and acts like a magnifying glass. And yeah, fun stuff. Thanks again, everybody. I'll see you.